what we want to do is I want y'all to understand what DNA does. Do y'all remember that it's instructions?
Yes, sir.
10 points. If you can tell me, what is DNA instructions for? It's instructions for making something in the cell. If you can tell me what it's instructions for, we already learned this. What is DNA instructions for making? It's for making something. DNA is instructions for making what? That's what we're going to learn about today.
Okay, hand it down to Trevor. Let him see it.
Yeah, there you go, Trevor.
What does DNA do? Okay.
Peach, correct.
No.
Blue, not right or wrong. Yes. Orange is correct.
We're getting there.
Is that 10 points?
Yes, purple correct. Anyone else? 10 points, yeah. White, no right or wrong. Green correct. Anyone else? It's instructions for making proteins. DNA is instructions for making proteins. And so I'm going to show you in the next 3 days of class that we have how DNA makes proteins. You remember what a protein is? A long chain of amino acids. This is code for making this. And so your essay on the next test is going to be, how does DNA make protein? That's about a 2-page essay. All right? Can you handle that? Not yet, but you hopefully will be able to after I teach it to you. So, it starts with a process called transcription. And here's a little picture. I don't have my PowerPoint made yet, but here's a little picture of how transcription works. But basically, during this process called transcription, can you say transcription?
Transcription.
All at the same time?
Transcription.
During this process called transcription, we take the DNA and we make a copy of a small part of it. That copy of a small part of DNA is called RNA. We make an RNA copy of the DNA chain. And it uses RNA nucleotides. Now, do you know where RNA nucleotides are found in the cell?
Yeah.
Go ahead and shout it out if you know it.
Nucleolus.
In the nucleolus. In the nucleolus, you have these RNA nucleotides. So imagine that this is the nucleolus. Can you imagine that this is the nucleolus?
Heck yeah.
And here's the DNA, which is a very long molecule. I only have a short segment, but you got to imagine it's real long and it goes all over the room here. Okay? And there's some other parts of the DNA. This part right here is called the coding region. That's what codes for the protein. This part here codes for the protein, but there's other parts of the DNA that are important too. This part is called the promoter. The promoter is where a special enzyme is going to attach. That enzyme is called RNA polymerase. Can you say RNA polymerase?
RNA polymerase.
Nice. An RNA polymerase attaches at the promoter, and then what it's going to do is going to slide past the operator. I'll talk about that in a minute. And it's going to unzip the DNA, and it's going to add RNA nucleotides one by one to copy the DNA. Click. This is my favorite part of teaching this. Click. I get to copy the DNA into RNA by taking RNA nucleotides and attaching them. Click.
Click.
Hold on, here's another one. Click. Here's another one. Click. Now, how do I know which one of these to grab and put on here? How do I know that? Am I just randomly doing it?
It's like the shape that
It's the shape that fits.
Because it's an enzyme.
Do it, Gracie.
Heck yeah.
It's got to be the right shape that fits. Now, if this DNA were TCAAGC, those are the letters these bases, could you tell me what the RNA bases would be? What goes with T?
A.
A. What goes with C?
G.
G. That's the rules. G always goes with C, A always goes with T. So if this is A, what would that be?
T.
You would think it would be T. But there's a special rule that you need to know about RNA. RNA doesn't have T. RNA has U instead of T. U is called uracil, and it's another base that we hadn't learned until now. RNA has uracil instead of thymine. That's the base that's right here. So T is not right. It's U. Wherever there's an A in the DNA, it connects to a U in the RNA. So that's a U. What is this next one? C. You see? That was hilarious. You see.
I get it.
Thank you.
I see it.
Hard to do science jokes with y'all. Yeah.
So if they're already thymines, we'll just put in an A.
If there's thymines, you get an A.
But they can't put it in themselves because we don't have it. Like,
Well, this RNA polymerase is just going to keep grabbing RNA nucleotides and putting them in. So wherever there's a T on the DNA, it'll put in an A. Wherever there's a C, it'll put in a G. Wherever there's a G, it'll put in a C. And wherever there's an A, it'll put in a U.
That's an interesting.
Does that make sense? Does that help answer your question?
yeah.
Oh, please. So this is a C, that's a G. And so what we've now done is we've made an RNA copy of the DNA chain. Oh, there's something else I got to tell you about these RNA nucleotides. The sugar is different than a DNA nucleotide. In an RNA nucleotide, the sugar is called ribose. Ribose sugar. That's why it's called RNA, because of the ribose sugar. Do y'all remember 10 points if you can tell me what the sugar is called in the DNA nucleotide? What's that called? 10-point question. It's called ribose in RNA. What's it called in DNA? 10-point question. Caitlin, are you with any of this? What are you doing over there? Taking notes? Look at you taking notes. Orange correct. Blue correct. Peach correct. Anyone else?
Who's that one?
You,
It's spelled wrong.
you could take notes by paper and use the notes on the quiz if we had one. White correct. Purple correct. Anyone else? Called deoxyribose. Deoxyribose. That's why it's called DNA, because it's deoxyribose sugar. So deoxyribose is the sugar in DNA. Ribose is the sugar in RNA. So there's two major differences between RNA and DNA. One, the sugar is different. Two, uracil instead of thymine. And a third difference is RNA only forms single strands. This thing's going to break away and it forms a molecule that is a single-stranded molecule. It's not double-stranded like the DNA. The DNA is going to go back together and you're back left with what you started with. And now the RNA, this is the copy of the DNA. This is called mRNA, or messenger RNA. It's the copy of the DNA strand, mRNA. That's what this thing's called. And it's a way of copying the DNA and taking it to this thing. You know what this thing is over here? It's a ribosome. Y'all remember ribosomes? They're on the rough ER. And what they do?
Protein.
They make protein. See, we're going to make a protein. Did I mention that at the beginning? The whole job of the DNA is to make proteins. So what we do is we copy the DNA and make this mRNA copy, and it's going to go to the ribosome, and that's where we're going to make the protein. The ribosome needs instructions on how to make the protein. That's what the mRNA is for. It's a message coming from the nucleus that goes out into the cytoplasm to the ribosome and tells it how to make the protein. That's what we're going to end up making.
Boom.
You see what I'm saying?
Yes.
Know what I mean? And by the way, we call this process of copying the DNA into mRNA, we call it transcription. Can you say transcription?
Transcription.
Wait, all at the same time?
Transcription.
Let's do it louder so Mr. Temple can hear us.
Transcription.
Okay, pretty good. I don't think you can hear it, but anyway. Okay. Oh, by the way, there's another site you need to know here. It's called the Terminator. I'm a Terminator. Did y'all see the movie Terminator? It was big in the late 80s.
Great. Yeah.
Might have been the mid-80s. Anyway, it was a fantastic movie. What happens is the RNA polymerase attaches at the promoter, it makes the mRNA copy, and then it falls off at the terminator. The terminator is the end of the gene. So we call this whole thing a gene. That's a gene. A gene consists of a promoter, an operator, a coding region, and a terminator.
So the polymerase falls off the DNA at the terminal.
At the terminator. Yes, McCall.
Does it attach to the promoter?
It attaches at the promoter side.
Does he have tape?
I have tape somewhere, I think. I don't know. What do you need tape for?
I wrote my page.
Oh, we'll try to find you some tape at the end here, okay? Other questions?
So it falls off, and then the DNA comes back together.
And the DNA comes back together. And you're left with your mRNA copy.
Wait, what does it do at the terminator?
At the what?
The terminator?
Polymerase.
The terminator is where the polymerase falls off. So it attaches at the promoter, it goes down like this, and makes the mRNA copy, and then it falls off at the terminator. Here's an example of a multiple choice question that might be on a quiz. At which site does the RNA polymerase attach to the DNA? A, the promoter, B, the operator, C, the coding region, D, the terminator.
Could you repeat the question?
At which site does the RNA polymerase attach to the DNA? A, promoter, B, operator, C, coding region, D, terminator. How many know that?
Wait. Promoter.
A, promoter, B, operator, C, coding region, D, terminator. 10 points if you can get it right. Just write it down.
Can you repeat it one more time?
At what site does the RNA polymerase attach to the DNA? A, promoter, B, operator, C, coding region, D, terminator. Just put A, B, C, or D.
Oh, can you put A for please?
Yeah. Orange correct. Purple correct. Green correct. You gotta get y'all some green paper. Or just call it yellow. Blue correct. Anyone else? A, B, C, or D.
Oh wait, A, B, C, or D.
Yeah, or whatever the name is. White correct. And peach incorrect. So, one more time. Listen up here. RNA polymerase attaches at the promoter. It goes down here and transcribes the gene, and then it falls off at the terminator. At which site does it fall off? Just yell it out.
Terminator.
The terminator. Attaches at the promoter, falls off at the terminator.
And the operator is another.
The operator, let's talk about the operator. The operator is an area, is a thing that can turn the gene off. There's a special protein called a repressor protein that can attach to the operator and turn the gene off. It's like an on-off switch.
Like a metabolic chain almost.
Wait, so what does
Or
the
saying-
operator do again?
The operator is the attachment site for a repressor protein, And when the repressor protein attaches to the operator, the gene is off. It's in what's called the off position. And that means the RNA polymerase cannot attach at the promoter if the repressor is there in the way. So it's a way, the operator is a site that is like an on-off switch. If a repressor protein is there, the gene is off. And if the repressor protein is removed, the gene is on, and you can transcribe the gene. Right now, in your cells, most of your genes are off. They're not functioning right now. Only the genes where when the cell needs to make something is it turned on where the cell can make something, where the cell can make a protein. What do you think about that, Skylar? Good stuff? What's your favorite part of the gene? It's got to be one of these 4 parts. Just pick one. Yeah, I like the operator too. Dawson, what's your favorite part?
Promoter.
Yeah? Okay, that's a good part. How about, does anyone like the Terminator? Best movie of the 80s. There we go. A couple fans of the Terminator. Let's talk about, oh, you want to see video footage?
Yeah.
Video footage of transcription? Here it is. Video footage of transcription.
Transcription is the process by which RNA is assembled from a DNA template. The 2 strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases along one strand. The portion of the DNA molecule that is a single gene or coding region is bounded by termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the 2 strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine.
It's making some mRNA, y'all.
This process ceases when RNA polymerase reaches the termination site.
Terminator.
The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
Huh? How cool was that?
So neat.
Wait, hold on. I got a question to ask about this. 10 points if you can tell me. Oh man, wait. 10 points if you can now tell me what the name of this blue thing is. What's the name of that blue thing? And another 10 if you can tell me what's the name of this brown thing. What's the name of this brown thing that's being made? 10 points for the blue thing, 10 points for the brown thing. And then I'll answer your question, Britton. Don't forget it.
I think that was my question.
Oh, okay.
Or part of it, at least.
Okay.
You agree, Trevor?
Yeah.
Blue, right on both. Orange, right on both. Orange, you glad I didn't say banana?
Yes, sir.
White, right on both. Peach, right on both. Anyone else? Yellow, green, right on 1. Purple, right on both. Is that everybody? So that's called RNA polymerase, and that's called mRNA. It's being copied from it. Question?
When the DNA or RNA polymerase puts like the RNA nucleotides onto the DNA after it splits it, are they just like, are they mRNA when they come off of it?
They're mRNA when they come off of it. When they're together.
But when they go on it, it's just RNA nucleotides?
Yeah.
Okay.
Do it, Chloe. Yes.
So the RNA polymerase separates.
It does. It
And
doesn't need
then
like a helicase
attaches.
or anything like that.
So
Yeah.
helicase is just out of it.
Yeah, there's no helicase in this one. Other questions? Y'all got to try to make sense of this in your heads because this is your next essay. Learn it now, don't wait till later. There's one more thing we have to talk about before lunch, and that's what we call mRNA processing. Let me get the light on for that so I can tell you what that is. This mRNA here isn't really ready to go to the ribosome yet. Oh, and by the way, I should probably draw a nuclear membrane with a nuclear pore. The mRNA has to come out through the nuclear pore. And so we have this mRNA strand. Right here, mRNA. But it's not ready to go yet. The first thing that has to be done is you have to add a cap to it. All mRNA wears a cap. It's called a guanine cap. And it's a special guanine. Special guanine. I thought I might have a special guanine, but I don't. It's a special guanine that's added to one end.
To the cytosine.
It's added to just on the very end here. I think it's the on the 5' end. You
I
add
get
a guanine.
So does it not matter what base it attaches to?
It doesn't matter. This could be whatever you transcribe. So you got to add a guanine cap on one end. I always thought about wearing a Georgia cap with a G on it, you know, when I'm teaching this, and I always forget to bring it. But and the people would ask me, "What are you wearing the Georgia cap?" And I'll say, "It's a G cap. I'll tell you about it in a minute." And it's a guanine cap. But once again, I forgot to wear it. But so anyway, it's got a guanine cap on one end, and on the other end, it has a what we call a tail, a polyadenine tail. And what that means is adenine after adenine after adenine is added to the back of it, like 200 adenines.
What?
And so every mRNA has this long tail to it. So what are these things for? This is what scientists are struggling to understand right now. They think the cap lets it get out of the nucleus.
Wait, so the tail is made
Of adenines, many adenines over and over and over, adenine nucleotides. The cap helps it get out of the nucleus, and the tail, they think, controls how long it lasts in the cytoplasm. If it has a long tail, it'll last for a long time outside the nucleus. If it has a short tail, it'll last for a short time before it's destroyed by enzymes in the cytoplasm.
So they're like the protection of the mRNA.
Yeah, that's what they think, but they don't know that. It's a hypothesis. It's not known fact yet.
Will it be
Does
on?
that determine what ribosome they go to?
No, they can go to any ribosome, but it does determine how many times, the length of the tail determines how many times it can go through the ribosome. The idea is that maybe every time it goes through a ribosome and makes a protein, one of the adenines gets knocked off. And so it goes through again, another adenine gets knocked off, and by the time you knock all the adenines off, it's done, it'll be destroyed.
Is that the tail?
That's what they might think about the tail, but they're not sure what that tail actually does. At least I haven't heard what it does. They might know now. I don't know. I've been teaching this for 20 years, and I've been saying that for 20 years. So maybe they know now. I might need to check on it. But anyway, they have to add a cap and a tail. And there's one more thing that has to be done to the mRNA. There's a special enzyme. It's got a cool name. It's called a spliceosome.
What?
Spliceosome. It's actually a group of enzymes that comes in, let's imagine it's my hand, a group of enzymes that comes in and cuts out little pieces of the mRNA and splices the other ends together.
Wait, what does it do again?
It comes in and it cuts out little pieces of the mRNA and splices the two ends together.
Kind of like trimming a hedge.
It's like trimming hedges. And what they think is that the mRNA has a lot of nucleotides that aren't necessary to make the protein, and so that these spliceosomes remove those unnecessary nucleotides.
So that's
To make the protein that it needs, you have to kind of give it a little trim and cut some things out. Yes?
So what does it mean when somebody slices their nucleus?
That means putting two things together and tying them together. That's splicing. So after we cut this out, we have to splice the two ends together. So splicing genes is putting two genes together to make one thing.
So is that before it leaves the nucleus?
Before it leaves the nucleus, all those have to be done. Cap, tail, and splicing.
The piece that's broken off, is that just like destroyed?
Yeah, it's just destroyed. You want to see a cool video showing you the splicing technique?
Yes.
This is an awesome video showing you how it splices. This is how it would really look inside a cell. This is a great video.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process...
So let me pause it right here. So this is the RNA right here, this long thing.
The yellow.
This is DNA that the RNA was transcribed from. This green piece is the piece that's going to get cut out. We call that an intron. Introns are the pieces that get cut out. These yellow things are the pieces that are going to remain. They're called exons. Introns get cut out and exons remain. Now let me back this thing up. Try it one more time without me interrupting.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one